I would like to then hand over to Karina from the Institute of Science at Gulbenkian Institute, who was also key in organizing this event. So thank you very much, Karina, for putting this together.
Thank you very much, Samuel. I'm Karina Xavier from the Gulbenkian Institute in Portugal, and I'm going to talk about the importance of the gut microbiota in protection against intestinal pathogens. So, as you've been hearing about today, the gut microbiota is important for many aspects of host health, ranging from nutrition, production of vitamins, or maturation of the immune system, and colonization resistance against the invasion of intestinal pathogens. And today I'll be focusing on this property of the microbiota called colonization resistance, which is basically the ability that the healthy microbiota community has to directly, through microbe-microbe interactions, act as a protective barrier against the invasion of pathogens such as Enterobacteriaceae like Salmonella or Clostridium difficile or like a Firmicutes Clostridium difficile or Vibrio cholera. In our laboratory, we use mice as an animal model to study the microbiota, and we look at how environmental perturbations such as diet or antibiotics perturb the microbiota, and to identify mechanisms that are required for recovery of microbiotic functions and microbiota specific bacterial members of the microbiota that can enable and accelerate this type of recovery. In untreated animals with a complex microbiota, as I said, they can provide colonization resistance against invasion of other bacteria. And in this case, what we're doing is giving a high load of Escherichia coli or E. coli to these animals that have an intact and untreated microbiota. And as you can see, 2 days after challenging these animals with E. coli, we can no longer see the presence of these bacteria in fecal samples. But this is very different from what happens if we first give the mice an antibiotic treatment. In this case, we were using streptomycin. We gave the mice streptomycin for 2 days and then challenged them with E. coli. And as you can see here in this plot, under these conditions, E. coli can colonize to very high levels. And because this E. coli strain is resistant to streptomycin, it will stay at very high loads throughout all the duration of the antibiotic treatment. So we can say this is an example of the type of experiments that show that antibiotic increases host susceptibility to pathogens, more specifically to Enterobacteriaceae. And as we've been talking about, antibiotics are still a great weapon and the best strategy we have to treat infectious diseases caused by bacteria. However, they do have this side effect of perturbing the microbiota. And what we typically see is that what's shown here is the composition of samples from untreated animals that have a very diverse microbiota composition, which is represented here by all these different colors. And upon streptomycin treatment, what we typically see is the loss of many bacterial sequences, leading to a microbiota composition that is much lower in diversity. And what happens is that animals with this type of high diverse microbiota composition have colonization resistant capacity, and these animals with this type of low diversity microbiota are very susceptible to the invasion of this Enterobacteriaceae. What we saw very early on when we start doing this type of experiments is that we saw a very high inter-individual variation between the response of the animals to the antibiotic treatment. In every case, we always see what I was talking about before, that we start with a very high diverse microbiota composition, and the antibiotics leads to a low diversity of microbiota. But the way the animals get to this to this low diverse microbiota stage can vary very significantly among animals. And we asked the question if this type of variation in response to antibiotics that we see in the animals would have a functional impact when we look at stopping antibiotics and how they recover from the antibiotic treatment, more specifically, how these animals would recover their colonization resistant capacity to pathogen. And in this experiment, what we did was to treat the animals for 2 weeks with antibiotics, then stopped antibiotics and allowed for a recovery period of 6 weeks, and then challenged the animals with E. coli. And again, we saw a very high inter-individual variation and how they respond to the E. coli challenge. Basically, how did they recover their colonization resistant capacities? There were some animals like these ones here on top, where E. coli was able to colonize to very high levels. So this means that these animals did not recover the colonization resistant capacity, but there were some other animals like this one here on the bottom that E. coli loads decreased quite rapidly, and they were 3 at the end of the experiment. They were about 3 orders of magnitude lower than in these other animals. So these ones, we could say that they could recover the colonization resistant capacity upon this recovery period after stopping antibiotics. We had some very interesting cohorts of animals that have very different phenotypes in terms of their colonization resistant capacities. And so then we could look and sequence samples, fecal samples from these animals, and look for sequences that could be associated with this colonization resistance to E. coli. And there was a specific sequence that was assigned to a sequence belonging to a Klebsiella genera, where it was always present in animals that were able to restore their colonization resistant capacity, but was always absent in the animals that did not have colonization resistant capacity. We plated the fecal samples from these animals in Enterobacteriaceae specific media, and we could detect the presence of this bacteria that we then performed whole genome sequence, and we could assign to a Klebsiella michiganensis, which is a non-pathogenic Klebsiella species that is present both in animals and in mice, in humans and in mice as part of their microbiota, and in our experiments was associated with the colonization resistance. So having isolated this bacteria, we could then test directly if it could indeed inhibit E. coli colonization. And in these animals here, after a recovery period, they were colonized with E. coli. These animals did not have this Klebsiella michiganensis. And in a group of animals here in blue, we challenged these animals with another E. coli species, and the levels of E. coli did not change. But when we challenged these other animals here in black with our recent isolate Klebsiella michiganensis, we could decrease and inhibit E. coli colonization. And we performed a similar experiment in germ-free animals, monocolonized with E. coli. E. coli could colonize to high levels, but when we introduced Klebsiella michiganensis, then E. coli inhibition was inhibited significantly. So Klebsiella michiganensis could inhibit E. coli colonization also in the absence of a native microbiota, indicating that the mechanism of colonization resistance of Klebsiella michiganensis is direct. We then gave the animals, these gnotto mice monocolonized with E. coli, a specific carbon source that was exclusively being used by E. coli and that Klebsiella michiganensis could not use, therefore giving a nutrition advantage to E. coli. When we did that, then when we challenged the animals with Klebsiella michiganensis, the inhibition of E. coli colonization was very mild. So this nutrition advantage to E. coli prevented Klebsiella michiganensis to outcompete E. coli, but this was then reverted when an E. coli mutant that was engineered not to use Galactitel was used. So in this case, again, we saw the inhibition. So we could conclude that Klebsiella michiganensis can inhibit E. coli colonization through a mechanism that we think is involving nutrition competition. So just to summarize a very long story short, this Klebsiella michiganensis is what we call a keystone microbiota that is part of the human and the mice microbiota. It's usually there at very low abundances. And what we could show is that this bacteria has a very diversified metabolic properties. And by having that, it can outcompete E. coli through nutrition competition and inhibit its colonization. And that Klebsiella michiganensis was sufficient to restore the antibiotic-induced dysbiosis towards and restore the colonization capacity after antibiotic-induced dysbiosis. And we showed this for E. coli, but also for Salmonella, and more recently with multi-drug resistant E. coli and Vibrio cholera. So as main take-home messages for today, I would like to highlight that what I told you is that a healthy microbiota can protect against Enterobacteriaceae pathogens, that a perturbed microbiota situation like a dysbiotic situation leads to an increase in susceptibility to infections. And the work I showed you today, I think it's a good example of the identification of specific members of the microbiota and exemplifying the hidden potential that exists in our microbiota communities. And in this case, I think it's essential to identify this type of microbes in order to establish strategies to restore microbiota following antibiotic treatments that can enable recovery of microbiota functions. So I think there's still a lot of microbes with amazing functions to be discovered. And this is just an example of one of them. With this, I'll just thank my group and Ana Rita Oliveira, who did most of this work. Thank you for listening.
Thanks a lot for this very good talk. Karina, are there questions? Please send them to the question and answer panel. There is one, but basically in Portuguese. So Karina, maybe you just want to read it yourself and translate for us others. Sorry about that.
Okay, so this Victor is asking about how can we better have the use of the antibiotics, and how can we have antibiotics that don't affect so much the microbiota, and how can we improve the regulations in countries where this is not so regulated. So there's a lot of questions here in this question. I would say that there are efforts for narrow spectrum antibiotics, but I think antibiotics will always lead to this increased to the selection for resistance. And I think we need to start using the antibiotics not only in the right way when they're really needed, but also in conjunction with other methodologies. So I think the future is really to combine the use of antibiotics with strategies that after the usage will enable restoring the microbiota. Yes, I think that would be my answer.
Thank you very much.